So good morning to all of you. Today our topic is spermatogenesis, the chapter human reproduction. So what is spermatogenesis? Spermatogenesis means formation of sperms. And as you know, formation of sperms uh, takes place inside the testis in the seminiferous tubules. So there are some animals who are known as who have breeding seasons, or they are seasonal breeders. So the animals which are known as seasonal breeders, testes become functional only in the season or breeding season. But the animals like human beings, testes are always functional. So there is no specific season or breeding season in case of human. But in case of some animals, there is a specific period when testes become functional in order to produce sperms in seminiferous tubules. So let's talk about spermatogenesis. As you know, it is formation of sperms inside the testis in seminiferous tubules. So this spermatogenesis process undergoes two steps. One step is formation of spermatids and second step is spermiogenesis. So there are two steps. First is formation of spermatids and then spermiogenesis. In the first step, which is formation of spermatids, this step actually includes three phases. One phase is multiplication phase, then growth phase, and the last one is maturation phase. Multiplication phase means here in this phase mitotic division takes place, mitosis takes place. That means uh, the cells get multiplied. Okay, in growth phase, that cell or that spermatogonia enlarges in size or increases in size, and this maturation phase, meiosis or meiotic division takes place in order to form spermatids and then sperms. So let's start. This first step is formation of spermatids with three phases. Multiplication phase means mitotic division, growth phase increase in size, and maturation phase means meiotic or meiosis or meiotic division. So first of all, as you know, in previous lectures uh, we had discussed about transection of testes, especially seminiferous tubules, and in the structure of seminiferous tubules we have seen a layer which is known as a germinal epithelium layer, and in the germinal epithelium layer there are two types of cells. uh, okay, so this is germinal epithelium layer, and in germinal epithelium layer there were two types of cells. One is germ cell and second one was Sertoli or nurse cell. And as you know, germ cells would undergo mitotic and then meiotic division in order to form sperms or spermatids. Okay. Actually, there is a difference between spermatids and sperms. Spermatids are actually circular in shape. They are non-motile. Okay. And sperms are motile in nature and have different shape to that of spermatids. And we would discuss in the next lecture. So this is germinal epithelium of seminiferous tubule. These are two types of cells, as you know, germ cells and these pyramid shaped Sertoli or nurse cells. So we would only talk about these germ cells because these germ cells now would undergo these three different phases and then this spermiogenesis in order to form sperms. Okay, so all of these are germ cells. Okay, we are not taking Sertoli cells right now. So all of these are germ cells. Out of this germ cell, we would take only one germ cell to make you understand. This is a one germ cell which is diploid. 2n is diploid. This germ cell would undergo the first phase of spermatogenesis, which is multiplication phase. Okay, so this one germ cell would undergo multiplication phase. Multiplication means all about mitosis. So mitotic division would take place. So this germ cell would become two daughter spermatogonia, two daughter circular spermatogonia, which are also diploid. Okay, this one germ cell would undergo mitotic division in the first phase, which is multiplication phase, to form these two daughter cells, also known as spermatogonia, which are circular non-motile spermatogonia. Okay, so these circular spermatogonia would undergo mitotic division again in order to form four spermatogonia. Okay, यानी एक जर्म सेल से दो बने, दो से बने चार. Okay, and from here to here, okay, तो यहाँ से लेके यहाँ तक is all about mitotic division or mitosis. Okay, or multiplication phase. So these four cells, these four circular cells which are diploid, are now known as these are spermatogonia diploid cells. Okay, and out of these cells, some cells actually would undergo a growth phase, then meiotic division in order to form spermatids, then sperms. And some cells or some spermatogonia would actually, they would behave like stem cells. That means when they would behave like stem cells, they keep on producing more spermatogonia, or they keep on dividing mitotically in order to form more spermatogonia. Okay, so out of these four, just for example, out of these four spermatogonia cells, these two cells would behave like stem cells. Okay, so that's why they are we can call them type A spermatogonia, and these type A spermatogonia, they would behave like stem cells. That means they will divide mitotically to produce more spermatogonia. That means they would keep on dividing. They would keep on dividing to form more spermatogonia. Okay, and these two cells, which are known as, or you can call them type B spermatogonia, would undergo the next phases. That means in growth. That means undergo growth phase. यानी दो cells behave करेंगे like stem cell in order to produce more spermatogonia, और कुछ एक cells या कुछ ये यहाँ पे जो हमने दिखाया है, ये दो cells, they would undergo the growth phase, then maturation phase. Okay, so यहाँ तो बात होगी, they would behave like stem cells and they would produce more spermatogonia under this mitotic division. हम बात करेंगे इनकी. So these type B spermatogonia would undergo next growth phase. Here you can see this type B spermatogonia would undergo growth phase. Growth means size would enlarge. Okay, size increases. So this type B spermatogonia now has increased its size, and it is also diploid. Why diploid? Because till now there is no meiotic or meiosis. There is no meiotic division have taken place till now. Okay, so that's why it is diploid right from here. Okay, so this enlarged or this increased or enlarged cell or spermatogonia, now known as primary spermatocyte. Okay, so when do we call this spermatogonia as primary spermatocyte? When this spermatogonia would undergo growth phase, and then when its size will be enlarged. Okay. So after becoming this primary spermatocyte, or after increases its size under this growth phase, this primary spermatocyte would undergo maturation phase, last phase. Okay, undergo maturation phase. And as I have told you, maturation phase means meiotic division or meiosis. So under this maturation phase, now you can see over here, this size has increased further. Okay, so this would undergo maturation phase. Now this, that means this primary spermatocyte would undergo meiotic division. Okay, so there are two meiotic divisions, meiosis 1 and meiosis 2. First of all, meiosis 1, which is reductional division. Okay, that means diploid cell now would become haploid. Okay, 2n now would become single n, 1n. So this diploid primary spermatocyte would undergo meiosis 1, maturation phase, would undergo meiosis 1, which is reductional division, would become this haploid secondary spermatocyte. Okay, these, okay, so meiosis 1, reductional division, this primary spermatocyte which was diploid in nature, after meiosis 1, this would become 2 haploid secondary spermatocyte. Now this is known as secondary, these are known as secondary spermatocyte. This is primary spermatocyte. After meiosis 1, after the reductional division, this would become haploid secondary spermatocyte. Okay, so after becoming haploid secondary spermatocyte, these secondary spermatocyte now would undergo meiosis 2. And meiosis 2 is equational division, like mitotic division. Okay, so they would undergo meiosis 2. Now these two cells now has become 4 in number, also haploid because it's all about meiosis. So these two cells now has become 4 in number, and these 4 cells now known as, after equational or meiosis 2, now these 4 cells now known as, these are haploid non-motile circular spermatids. Okay, so these are known as haploid non-motile circular spermatids, and these spermatids would undergo the process named spermiogenesis. So under the process of spermiogenesis, these 4 haploid circular spermatids would change into these motile sperms. Okay, so it's all about spermatogenesis. These 3 phases: multiplication phase, growth phase, maturation phase, mitosis, increase in size, and meiosis. Then this simply spermiogenesis means change of these spermatids into sperms. In the next lecture, we would discuss about the structure of sperm and how these spermatids would change into sperms. Okay, so we would discuss all about the change of spermatids into sperms and the structure of sperms in the next lecture. Thank you very much.